Hey folks, I'm going to finish up our material on angiosperm reproduction today. I'm going to start with a quick review, looking back at this question about carpels versus pistils. So remember that the female reproductive world, the gynecium, is made up of carpels, and those can either be separate from each other, or what we call a simple pistil, where the gynecium is made up of one carpel. Or they can be fused together, where the multiple carpels are fused during development, and we call that a compound pistil. I showed you good examples of a compound pistil. I did not show you a good example of a simple pistil. So I mentioned we could have one simple pistil, and that's the situation that we see in like a bean or a cherry. The gynecium of those plants are made up of one simple pistil. But we can also have many simple pistils where they're not fused to each other. And I didn't have a good image of that. And so I pulled this together. This is actually a strawberry flower. And what we see in the center of this strawberry flower is, we can see this, we can't see the sepals. Here's one sepal poking out here. We see the petals are white. And then we see the andrecium, the male reproductive whorl, made up of these stamens. So we see all of the stamens around. And then in the center is the female reproductive whorl, or the gynecium. And the gynecium here is made up of multiple separate, many separate simple pistils. And so that's what these are, all arranged on sort of a dome-shaped or conical-shaped receptacle, where they're all arranged. So if I do a section of this, if I cut this through in a longitudinal section, this little inset picture shows that here. And so here, this is all receptacle tissue in the center here that these pistils are, each of these separate pistils are arranged on, and each separate pistil, each one of these is a separate pistil with its own ovary, its own style, and its own stigma. And so pollen will be deposited on each of those. Each one of these ovaries has one ovule inside of it. And that ovule has an egg cell. That egg will get fertilized, and this will develop into the fruit. And so the ovary, so this will have many fruits. Now, this is different than what we think of as a strawberry fruit, right? When you think of a strawberry fruit, you think of that big red thing with the black dots on the outside of it. Well, it turns out that big red thing, that fruit part that we eat, is actually receptacle tissue. That's all this that gets swollen and juicy. The fruits are actually, the ovaries develop into those little black, what we think of as the strawberry seeds. Those are not seeds, those are a single-seeded, hard little fruit. And so strawberries are a group of fruits that are aggregated together on that fleshy receptacle. We'll come back to the fruit stuff, but I wanted to make sure I showed you a good image of multiple separate simple pistils, each one made up of one carpel. So this gynecium is made up of hundreds of carpels, but they're separate from each other and not fused, and they're all arranged on this sort of enlarged receptacle tissue. Okay, cool. So we talked through that. We talked through different types of flower terminology. And what I want to do now is really dive into the details of gametophyte development and the gametophyte stage in angiosperms. And so just to do that, we got to remind ourselves of the life cycle of lamb plants. And remember that the life cycle of lamb plants follows the alternation of generations. And that alternation of generations proceeds alternating between this haploid gametophyte stage of the life cycle that produces gametophytes, or that produces, gametes, and the diploid sporophyte part of the life cycle that produces spores from meiosis. And remember the alternation between those two stages in the life cycle. And so this is a example of a horsetail fern showing you the separate sporophyte and the separate independent gametophyte generation in these. And in this case, these were homosporous. where we just have one type of spore. That develops into the gametophyte and onward through the alternation of generations. Well, angiosperms, if you recall, are heterosperous. And so that is an important distinction to be made between that and some of our other land plant lineages. And so what was heterosperous? Well, heterosperous is that we have two types of spores. And specifically, we have male microspores and female megaspores. And whenever we have heterospory, remember that the development of the gametophyte stage is much reduced, and the development of the gametophyte is endosporic. And so these microspores and megaspores develop into endosporic gametophytes. What does that mean? Well, that means that the spore, the gametophyte stage of the life cycle, both the male gametophyte and the female gametophyte, happens within the spore wall. So inside of the spore wall. And so as such, everything is very reduced. When we looked at this fern life cycle, the development of that gametophyte actually happened outside of the spore, right? The spore germinates and grows into this multicellular haploid gametophyte. That in this case, these are independent, and then through after fertilization of the gametes, we then move into the sporophyte stage. Well, here the gametophytes actually develop inside of the spore walls. So because we're heterosperous, we have to talk about meiosis in two different places. Meiosis meiosis happens in both the anthers of the flower as well as inside of the ovule of the flower. Those are the two places where meiosis occurs, and the two places where then those separate endosporic gametophytes are going to develop. And so let's just look at the anther side of this to start with. Meiosis occurs. That microspore that produces the microspores, and those microspores then develop into a mature gametophyte, male gametophyte, and that is what we call the pollen grain. And so the pollen grain itself is the male gametophyte, the microgametophyte, is inside of the pollen grain. On the female side, inside of the ovary, we have ovules, and inside of that ovule is where meiosis occurs. And we form megaspores after that meiotic event. And I'll go through the details of this again, so I'm just going to brush over it here. But we have a megaspore inside of that ovule, and then through mitotic divisions, we develop into the multicellular female gametophyte. And so in the case of the male microgametophyte, that was the pollen grain. In case of the female gametophyte, it's something that we call the embryo sac. And so the embryo sac itself is the female gametophyte. It, in angiosperms, as we'll see, it consists of 7 cells and 8 nuclei. That's the whole gametophyte, female gametophyte. In the case of the male gametophyte, we end up with 2 or 3 cells, but 3 cells is the gametophyte, a tube cell, generative cell that divides into 2 sperm cells, and so we end up with these 2 cells and 3 nuclei in the case of the male gametophyte. So everything is very reduced, and it's happening inside of the spore wall. Okay, so we still have multicellular haploid gametophyte stages. They're just very reduced inside the spore walls because of the endosporic nature of this heterosperous plants. Okay, the other thing I want to point out on here is then we need to have the male gametophyte get close to the female gametophyte, and that is what we call pollination. So pollination can be defined as the movement of the male gametophyte close to the female gametophyte. And this can happen in a number of ways. So this can happen through wind. So abiotically, this can happen through water. This can happen also through biotic interactions with pollinators like other insects or birds or even mammals in some cases. And I'm actually going to have you, for our last part of the angiosperm reproduction section of the course, we're going to look at the ecology of pollination through an awesome video called Sexual Encounters of the Floral Kind. And so I'll post that, and I've got some note sheets for that as well. It's a really cool old video on pollination biology and all the crazy things that plant sex lives entail. So anyway, we'll talk about pollination, but pollination is the movement of the male gametophyte close to the female gametophyte. And then fertilization is actually something we're familiar with, the fusion of sperm and egg. Pollination does not always result in fertilization. Successful pollination will result in fertilization, but it doesn't always result in fertilization. So there is a distinction between the movement of the male gametophyte to the vicinity of the female gametophyte, or pollination, and the process of fertilization, transitioning between the gametophyte and sporophyte generations in angiosperms, or in seed plants where we have pollen as our male gametophyte. Okay, so we've got these heterosperous organisms, these heterosperous plants that are making both male and female endosporic gametophytes. And we'll go through the details now about the evolution of these different structures, the male reproductive parts and female reproductive parts, and then the development of these gametophytes. So just as a reminder that the synapomorphy for angiosperms, one of the synapomorphies for angiosperms is the flower, and specifically it's the stamen, the part of the andrecium, the anthers and filaments, and the carpel, or the part of the gynecium. Those are really the synapomorphies. The synapomorphy of a flower is the presence of these male and female reproductive parts. And so the first thing I want to look at is the evolution of the stamen, and just kind of get an idea of where the stamen came from, and a little bit about its morphology. And so, if just as a reminder, stamens are made up of the anthers and the filaments. And if we go ahead and we take a cross section of the anther, remember, is the site of where meiosis is occurring. And so this is really, these are sporangia, right? These are where meiosis occurs to make spores. These are just modified sporangia. And when we look at a cross section of that and look at it with the microscope, before meiosis occurs, we have microspore mother cells. And those microspore mother cells are surrounded by what we call a tapetum. And the tapetum is a nutritive layer for the developing male gametophytes. Or pollen grains. And so before meiosis occurs, we have that mother cells. Those mother cells, these are all diploid cells in this slide right now. And the tapetum is made up of diploid sporophyte cells. So these are all sporophyte cells. And then meiosis occurs, and that's our transition. Meiosis, remember, is our transition between our gametophyte and our sporophyte and our gametophyte generations. And so once meiosis occurs, then meiosis develops into those megaspore mother cells undergo meiosis, and we create our spores. And then inside of the spores, the male gametophytes develop, and so those would be the pollen grains. And so these sacs, the anther sacs themselves, where those sporangia, where meiosis is occurring, and then our endosporic gametophytes develop inside of the spore wall of each of these microspores, developing into that male gametophyte. So where did these things come from? If we look at through the fossil record and through and look at the variety of stamens that we see throughout angiosperms, we actually can interpret the evolution of the stamen as a modified microsporophyll. So a sporophyll is just a fancy word for a fertile leaf. And this fertile leaf would have had 4 microsporangia on it. And so here we see sorry, this scanned in pretty poorly. We see sort of a modified leaf, a fertile leaf with sporangia on it. And we can imagine that over time, we've had the reduction of this structure down to the filament and just the anther sacs, and that is the evolution of the stamen itself. And so we go from it's basically a modified microsporophyll that has these 4 microsporangia on them, and we have this reduction. And we look at some of our land plants, some of our angiosperms today actually have stamens that look very much like this, that are flattened out with long, elongated marginal anther sacs, and we can see this progression from this modified leaf to what we see as a modern day stamen. And we see that both in the fossil record and, like I said, in the diversity of stamens that we see throughout angiosperms as well. Okay, on the other side, we've got the carpel, which is the part of the gynecium. And so where does the carpel come from? Well, if we look at a cross-section of a carpel, we think of it like I described with that strawberry example at the beginning. This carpel will mature into a fruit, and the ovule is going to mature into our seeds inside of that. And if we do a cross-section, maybe this is a cross-section through the ovary part of a single carpel, one of the things that we see with this is that there's both an external and an internal epidermis. So we can find the epidermal layer. Epidermal layers, remember, are typically between the inside of a leaf or a body plan and the outside environment. But this is actually a cross-section through an ovary. And so we're looking through that ovary and looking down inside of it. And what we see is that even on the inside of the ovary wall, there's an internal epidermis layer. So why is that? Why is that the case is really what we're looking at. Other thing I want to mention, just because I have it on here, is one of the other synapomorphies for angiosperms is that each ovule is surrounded by 2 integuments. So we call these bitegmic ovules. Those 2 integuments are just as protective layers that are surrounding the outside of where the female gametophyte is developing inside of that megaspore wall. I'll go through that development separately. The main thing I want to show you here is in the cross-section of the carpel, we have both an external and an internal epidermis. So what's the deal with that? Well, if we think about the evolution of a carpel in the same way that we thought about the evolution of a stamen, the evolution of a carpel comes from a modified megasporophyll, so a fertile leaf that contains megasporangia. And so in the fossil record, we can actually see what we call these conduplicate carpels. Conduplicate carpels are really loosely fused carpels that came from this modified leaf. And so this leaf basically had sporangia, megasporangia along the margins of it, and then this leaf rolled up on itself. And when it rolls up and seals, now that becomes an enclosed structure, that ovary with the ovules on the inside of the carpel. And so what does this have to do with the inner and outer epidermis? Well, if this is a leaf, it's got an epidermis on the upper surface and on the lower surface. When this curls in, now I've got an inner epidermis and an outer epidermis. So this is really good evidence that these are modified leaves, that ovaries, carpels, are modified leaves that have rolled up on each other developmentally and are now enclosed as ovules on the inside. This creates the clothed seed that we see. Clothed seed, remember, is what angiosperm means. the clothed seed, the seeds developing inside of a fruit. That is the ovules inside of the ovary, the ovules inside of the carpel of the gynecium. Okay, I forgot that I had this slide in here. Actually, this shows the carpel situation pretty well. So carpels can be free, meaning not fused to each other, or fused to each other. And so when they're free, this is what we call that simple pistil. And then fused, we call the compound pistil. And so here's a couple of examples showing the free carpels of this raspberry plant. Raspberries are related to strawberries, coincidentally. These also have many separate simple pistils, each one developing into what we think of as raspberry fruit. Well, raspberry is actually a whole group of fruits. Each one of those little round balls is one of the developing fruits from one of these ovaries in a raspberry. Here's a tamarind. This is in the legume family. And we see one simple pistil, one made up of one carpel. And then papaya, an example of a fused a compound pistil where we have one ovary and we've got these 5 separate styles and stigmas, so that's 5 fused carpels. And to get from a simple pistil to a fused carpel, we also have fossil evidence for this. Another crappy scanned slide, I'm sorry. But what we see here is we see sort of the situation showing a single fused carpel, that rolling up of that modified that sporophyll or modified leaf. If we have several of these arranged near each other on an axis, we can imagine them fusing together. And so here's an example of 3 fused carpels and how that would look. And so each one of these is the separate carpel that's been fused together developmentally. Okay, so like I said, there's fossil evidence for these sort of intermediates in both stamen and carpel development. And so if we look back at the fossil record, you remember this slide from the beginning of, I think from the first lecture. What I want to point out here is this fossil. So there's a fossil out there. This was found in China called Archifructus. It's about 140 million years old. And has been touted as the first flower. There's actually an incredibly good documentary on this called First Flower on PBS. I don't remember if it's a Nova special or not, but it's on PBS and you can find this. So if you were interested in sort of the fossil paleobotany side of this, check that out. This is a cool documentary about Archifructus and other first flowers from these deposits in China. But if you notice, Archifructus has this very elongate axis. And really what that is, I'll show you a diagram, sort of drawing, artistic reconstruction of this first flower. What we see is that really what this contains is carpels and stamens. There's no perianth, no petals or sepals associated with this. We've got no perianth. And we just have carpels and stamens. Sorry if I said sepals. So that's on this sort of elongated axis. And so the idea is that over time, this axis is compressed. And so we just kind of get compression of this axis into a modern day flower with its 4 whorls, the 2 perianth whorls, and then the 2 fertile whorls with the andrecium and the gynecium. But this is about 140 million years old. That's our first evidence of first macrofossil for angiosperms. We do have pollen fossils that go back a little beyond that. Okay, so let's now move into thinking about gametophyte development and how are the gametophytes actually formed in plants. And so if you remember, there are 2 places where in the plant where meiosis is going to occur. Meiosis is going to occur inside those sporangia in the pollen sacs, so in the anthers. and then it's going to occur inside of the ovule, the developing ovule, in the inside of the ovary of the flower. So both of meiosis is happening inside the flower. The majority of the flower then is sporophyte tissue, and it's inside of the anther sac and inside of the ovule, inside of the ovary, where meiosis is actually occurring. So let's start with the female gametophyte. And so we'll look inside of that ovary, and we see a megasporocyte, or that's another word for a megaspore mother cell. So this is a diploid cell that's going to undergo meiosis. That's our transition between, between the gametophyte and the sporophyte and gametophyte stage. Megaspore mother cell undergoes meiosis. And so just because we can look at this sort of, we have these drawings, but I'm going to go back and forth between these diagrams and these actual cell prepared slides of the cross sections of these. So here's an ovary cross section, and we can look and see an ovule in section and see, or developing ovule, and see that megaspore mother cell as part of that development. Now, megaspore mother cell also is surrounded by the two integuments. So remember, this makes the bitegmic ovule. It's surrounded by those two integuments, and surrounded by the nucellus. And just like in the male side of this, there's a nutritive tissue in the female side of this, in the development of the female gametophyte, and that's the nucellus. And so this is nutritive tissue for the developing female gametophyte. Okay, so that's there's the megaspore mother cell. This will undergo meiosis. And that meiotic event will result in that meiotic, one thing I forgot to point out on here is the micropyle. Micropyle is the opening for, it's the opening for the pollen tube. For pollination to occur, is that micropyle. So there's that little opening there. Okay, sorry about that. Let's look at the process for the formation of that female gametophyte. So we have meiosis occur in the megaspore mother cell. That creates 4 megaspores. 3 of these degenerate and go away, and we have 1 megaspore mother cell that is maintained. This megaspore mother cell then undergoes mitosis and develops into the female gametophyte. And so a few rounds of mitosis, and we create these 8 nuclei. And those 8 nuclei then develop into the mature embryo sac, and the embryo sac is the name for the female gametophyte. We end up with 7 cells that are formed, and one of those cells has 2 nuclei, these 2 polar nuclei. So the mature female gametophyte in an angiosperm is made up of 7 cells and 8 nuclei. And so let's just take a look at that embryo sac. And so here's 1 megaspore produced by meiosis that undergoes 3 rounds of mitosis to create the 7 cells and 8 nuclei female gametophyte. And so here's the female gametophyte embryo sac surrounded by its integument layers. And we see our cells are all arranged here. Can't see quite all of them. There's 3, 4, 5, 6, 7, and then another nuclei that's out of focus here. We end up with our 7 cells and 8 nuclei in that embryo sac. Here's a better picture of those. 7 cells and 8 nuclei. A key to this is that these are very reduced female gametophytes. This is the first place where we see no archegonia. So the egg is not contained in an archegonium, but rather we end up with the egg cell, 2 cells that we call synergids, two polar nuclei that become important. In just a minute, you'll see that. And then three cells on one end of the embryo sac called the antipodals. The synergids, the egg, and the polar nuclei all have functions that we'll talk about, but the antipodals not so much. So when it comes to pollination, we'll talk about these different cells. Okay. So that's the development of the female gametophyte or the embryo sac. Let's now take a look at the development of the male gametophyte or the pollen grain. Male gametophyte, this one's a little bit simpler. And so inside of that microsporangium or the anther sac, we have microsporocytes. These are the microspore mother cells. Those are diploid and they undergo meiosis to transition to that gametophyte stage. Those 4 microspores, the meiosis will produce 4 microspores, and each microspore will undergo mitosis to create, so each microspore undergoes around a mitosis to create a generative cell, a tube cell, and these are all surrounded by the spore wall, this tough sporopollenin impregnated spore wall, and that's a mature gametophyte. So the mature male gametophyte here has 2 cells, the generative cell and the tube cell, and those are all surrounded by the tough outer coat of that spore wall. So let's look at these for real. So we see our microspore mother cells here. Those undergo meiosis to form our pollen, our tetrad of microspores. So there's our 4 microspores that were formed by meiosis. If we look inside of any one of those, as that male gametophyte matures, we end up with these 2 nuclei. A generative cell, this generative nucleus here, and the tube cell, the rest of it with the tube nucleus here. That generative cell will then undergo one more round of mitosis, and it divides into 2 sperm nuclei. So the generative cell divides and forms the 2 sperm nuclei. These nuclei we call them sperm nuclei. They're sperm, the nucleus, the genetic material, but they actually don't have, they're not motile sperm. So these don't swim. These are just 2 nuclei, 2 sperm nuclei. Those are our gametes in there. So now we've got this mature male gametophyte with its 2 sperm nuclei and its tube nucleus. Once pollination occurs, the pollen grain is moved to the stigma of the flower, of the ovary, the gynecium of the female plant, and a pollen tube. That tube cell develops into a pollen tube, and that pollen tube actually grows down through the style of the pistil of the gynecium and toward the ovule. And as that grows down, that tube nucleus moves along with it, but then also the sperm nuclei, those 2 sperm nuclei move down the tube. And so the pollen tube actually is a delivery mechanism for the sperm nuclei, for the sperm to get to the ovule, to the embryo sac, to that female gametophyte. And once that moves to the female gametophyte, remember, that is pollination. So pollination is the movement of that pollen grain, the male gametophyte, to near the female gametophyte. And that pollen tube then is going to grow down through the style. That pollen tube is going to grow down through the style and actually into the near the female gametophyte. And there's really cool images of pollen tubes growing. So here's a couple pollen tubes, some pollen on the surface of a stigma, and we see those pollen tubes growing through the style, and the pollen tubes will grow all the way through the style and out into each one of the ovules. And so each one of these is an ovule. We see our pollen tubes growing in, and that delivers these sperm nuclei to the female gametophyte. And another of the synapomorphies for angiosperms happens here, and that is actually the process of double fertilization. And so there are two sperm nuclei. One of those sperm nuclei fuses with the egg to form the zygote. The second sperm nucleus fuses with our two polar nuclei, these two in the middle here, fuses with these two polar nuclei to form the triploid endosperm. So 3N. And this triploid endosperm is nutrition for a developing embryo. So double fertilization resulting in triploid endosperm was one of our synapomorphies for angiosperms, and that's this process. So that pollen tube grows down, it grows in through the micropyle and actually grows into a synergid. So the function of one of the synergids is to direct pollen tube growth. The function of the egg is to form the zygote, and the function of the polar nuclei are to form the triploid endosperm, this nutritive tissue for the developing embryo. So if we take just a few different diagrams of that to show you this double fertilization, we've got our two sperm nuclei moving through a pollen tube, that tube growing into a synergid. One of those nuclei is going to, the sperm nuclei is going to fuse with the egg to form the zygote. And the second sperm nucleus then is going to fuse with these two polar nuclei to create the triploid endosperm. And so just see that in a little bit of a different view. We've got egg plus the sperm. That's going to create the zygote. And then we have our two polar nuclei plus that other sperm cell, and this creates our triploid endosperm. Oh, I have that written right here. So the polar nuclei plus the sperm nucleus makes triploid endosperm, which is that nutritive tissue for the developing embryo. So that brings us to embryo development. And so remember that seed plants, land plants are also called embryophytes. These are things that develop an embryo, a multicellular diploid stage. This is the start of the sporophyte stage, and we end up with our endosperm inside of that seed. So as this fertilization occurs, this now ovule develops into a seed. The seed contains the embryo as well as all of that endosperm or that nutritive tissue. And so let's just take a quick look at some seeds versus fruits, and we'll look at a few different types of fruit types. But this, I think, shows this quite well when you look at a peanut. A peanut actually demonstrates all of this perfectly. So there's a few cool things. One is that inside of the peanut, we can see the embryo. Oh, I can't spell. Embryo is inside, that's that little heart of the peanut, that's the seed. Inside of that fertilized ovule, we have that embryo develop. And that embryo develops from all of this triploid nutritive tissue. The food stores, or that triploid endosperm that's in there. When you are, so the seed part of the a peanut, the seed is the part that we eat. That papery layer that's on the outside, well, that papery layer is actually the integuments. And so that brown papery layer out there, that's our protective tissue for that developing developing seed. So we've got this outer coat, those integuments on there. That's our seed coat. And we have our dormant embryo. So we got all those pieces inside there that we see. So the seeds are anything that comes from a fertilized ovule, whereas a fruit is the structure that's derived from carpel tissue or from the ovary itself. And so in this case, the fruit is the ovary wall is the outside shell of the peanut. And so a peanut is a fruit. Lo and behold, nut is a fruit type. Peanuts are a fruit. Inside of there are two seeds. Most of the time there are two seeds inside of those peanuts. So that would have been an ovary with two ovules inside of it, if that makes sense, or a carpel with two ovules inside of it. So let's look at a few others. I already blew this one before. I already told you that when you eat a strawberry, you're actually eating fleshy receptacle tissue, and that the fruits of the strawberries are these little tiny black dots that get stuck in your teeth. Those are what's derived from the carpel tissue, from these multiple separate simple pistils arranged on this conical or dome-shaped receptacle, not fleshy receptacle. So the fruit equals the black, I'll put seeds in quotation marks. What these actually are is single-seeded, hard fruits. We call them indehiscent fruits. They're fruits that don't open up at maturity. I'm not too worried about that terminology, but these are actually a fruit type called a nakeen. And that's a single-seeded, hard, indehiscent fruit. These are all arranged on a nice, juicy, fleshy receptacle. So what about when we eat an apple? Well, when you eat an apple, how many carpels do you think this came from? 1, 2, 3, 4, 5. Look at that. There's 5 chambers inside of there. This is a compound pistil. Apples are from compound pistil with 5 fused carpels. And actually, the ovary, that like membrane part of the inside core of the apple that also gets stuck between your teeth, that's actually the ovary wall. And so the fruit is actually just the apple core itself. And the part of the apple that we eat is the fleshy receptacle again. So the receptacle actually grows up around the apple, swells up around the fruit part, the core of the apple. And it's the fleshy receptacle that we're eating. And so in class, had I been in class with you, I would have had an apple prepared and said, who likes apple fruits? And someone would raise their hand, and I would then push the core of the apple out, and I would give that part to the person who likes the apple fruits, and I would take a big bite out of a fleshy receptacle and enjoy that fleshy receptacle. Because that's the fruit part is actually the part we discard. Okay, another one of my favorites to talk about with fruits. This is the favorite mealtime conversations around my house is whether these are fruits or vegetables and whether they're seeds or fruits or what part of the plant we're actually eating. Super fun for me. So coconuts are actually are the world's largest seed. The part of the coconut that we eat is the seed part of the coconut, and it gets cooler than that. And so the hard part on there is our seed coat, that hard seed coat. The flesh and the milk of the coconut is actually endosperm, and so that's that triploid nutritive tissue for that developing embryo. It gets even cooler because coconuts have both cellularized endosperm, and so that's the white flesh that we eat, but the milk is actually free nuclear endosperm, and so these are just a bunch of nuclei floating around inside of there. triploid nuclei. 3 nuclear endosperm is really what the milk is that you eat and drink in a coconut. That's awesome. And the fruit part of the coconut is actually the fibrous husk. And so a lot of times people haven't seen these because you don't buy this part in the store, but coconut seeds are surrounded by this fibrous husk, and that's derived from the carpel wall, the ovary wall, or the carpel, and that's for dispersal. So coconuts disperse by floating on the water. They get into the ocean, that fibrous husk allows them to float. They have this huge store of energy inside of that seed. These wash up onto a beach, and the seed will germinate and eat up that endosperm for the development of that seedling. Okay, last one here are my favorite fruits and fruits that rule the world, and those are grains. And so all of the grains that we eat, whether they're corn or rice or wheat, these are each one of those corn kernels, each one of those grains of rice, each one of the grains of wheat are all an individual fruit. These are all single-seeded fruits where the fruit wall and the seed wall are fused to each other. It's a special fruit type called a caryopsis, but they are fruits. And so all of the grains, when you're eating your rice or eating your bread, you're actually getting your fruit intake as well. Okay, I'm going to go ahead and stop us there. That wraps us up with this material. I'll post one more bit on pollination ecology, as I said, a link to a separate video, and that will wrap up our angiosperm reproduction section. Okay, thanks a lot, and I'll see you guys later.